So, what is the physical seat of thought? What is the source of our emotions, our decision making, our passions, our pains, and everything else? Well, it's the brain, and it's said to be the most complex mechanism in the known universe. You might accept, given all it is and given all it does, that will look very pretty, Philips swimming lights and glass tubes and mysterious colors. But in fact, it looked really kind of gross. It looked at the 3-day-old meatloaf. It's gray when you take it out of the head, and inside the head is bright red because of the blood. In fact, it turns out very surprisingly that the source of our mental life is meat. In fact, you could eat it. People have eaten brains. I've had brains with cream sauce, not human brain, mind you, but I've had brain with cream sauce. It's not bad, but it makes the puzzle all the more harder. How can this fleshly thing give rise to mental life? That's the question I want to explore in this lecture and the rest of the lectures. I want to do so by starting with the smallest relevant parts, different parts of neurons. Then explore how the neurons are connecting together, how they're wired up, how they form different parts of the brain, like the hypothalamus and the frontal lobe. Finally, talking about the brain and the larger perspective, looking at the two halves of the brain, the left half and the right half, and how they interact. Now there is a lot of stuff in the brain, a lot of chemical stuff, lots of different parts, but where the action is, the part that does the thinking, the part that is the focus of most of our research, is the neurons. It's not an accident they call the study of the biological basics of thought neuroscience, because it all comes from the neurons. So you can see here pictures of neurons interacting together. Here's a diagram that depicts a typical neuron. So what you see is the dendriters, and dendriters receive signals from other neurons, either excitatory like pluses or inhibitory minuses. Then they get to be the cell body, which sums up the pluses and minuses. When you research a certain threshold, a certain amount of pluses, there is neuron firing. Firing takes place within the axon, and the axon is much longer than the dendriters. In fact, for some motor neurons, it's very long indeed. This axon is running from your spinal cord all the way to your big toe. You could think of the relative sizes of things in terms of a basketball and the 40 miles garden hose. Surrounding the axon is what's called a myelin sheath. The myelin sheath is, you can think of it as insulation, as a like insulation on a wire. So the information comes through the dendriters and sums up in the cell body, and it's transmitted through the axon. So what neurons do is they sum up, they transmit information, and we know that there are a lot of them. By some estimates it's 100 billion, or the estimate is 10 to be different and very rough, but there are billions upon billions of neurons, and each connect to thousands, maybe tens of thousands of other neurons. So the fact that you have something of this degree this degree of structure which there's no way to replicate in any machine, the numbers are just too big, is why people might describe the brain as the most complicated machine in the universe. At least this is funny. It's made of meat maybe, which is kind of disappointing, but at least it shows its incredible internal structure. So, neurons come in three flavors. There are sensory neurons, which take in information from the environment, from the external world. There is motor neurons, which go from the brain out to your motor control. So, if you touch something hot and if you feel the pain, that is sensory neurons. If you rent your hand back or you reach for something, that's motor neurons. Finally, there's interneurons, which connect different neurons without making contact with external world, either thought sensation of thought motor action. Now the main thing to think about for neurons and neuron firing, and that is a all of nothing or all nothing. It's like firing a gun or sneezing. Neurons either fire or not, they don't. How you might think that's a little bit strange, particularly when you think about sensory neurons, because your experience seems to be a continuum. So you have sensory neurons in your eyes, and you can decide from a very dim light and a very bright light. You have sensory neurons in your fingers, and you could distinguish between gently touching something versus being stabbed on the tip of your finger or something. But still the neurons are all or nothing. The way we get to this continuity of experience is that neurons can code for instead in different ways. So one way is in terms of the number of neurons that fire. If X neurons corresponds to a mind experience, X times 10 neurons may correspond to an instance experience. Another factor is the impulse frequency of individual neurons. An individual neuron might denote a mild sensation by going fire, 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 fire. Well, it might denote an intense situation with fire, 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 fire. So you have neurons and the neuron talk to each other. They talk to each other because axons, an axon of one neuron will communicate with the dendriters of another neuron. A long time ago, people used to think that neurons were wired up together like a computer, but in fact, neurons don't actually touch another. There is a gap between the axon terminal of on one neuron and the dendriter of another one. A very tiny gap, typically of like 1 to 110,000 of a meter wide. This gap is known as synapse. When one neuron fires, the axon releases neurotransmitters. There are chemicals then shot out over the gap and affect dendriters and other neurons. As I said before, the effects of these neurotransmitters could be excitatory, which is that they raise the energy, so they increase the likelihood of a neuron firing, or inhibitory, so that they bring down the likelihood of neuron firing. What's interesting is that different neurons shoot out different neurotransmitters, so they have different effects on other neurons that they made contact with. In fact, a lot of psychopharmacology, both intends to cure various psychological or physical diseases by giving medicines, or recreational psychopharmacology designed to increase pleasure of different forms, or sometimes help people work or help people focus. Work by fighting with the neurotransmitters, and this can be either antagonists, and they lower down intensity of things by binding to the dendriters, making it hard to create more neurotransmitters, or they can increase the amount of neurotransmitters available in different ways agonists. So you're either pumping up the volume or turns down the volume. So you think about different drugs and their effects. There are curare. Curare is a drug that used by South American Indians. It's an antagonist. It blocks motor neurons from affect their muscle fibers. It keeps your motor neurons from working, and that it does, it's paralyzed you, and a large enough does, it kills you, because motor neurons all keep your heart beating. So shut that down and you die. There is alcohol. Now alcohol also has an inhibitory effect. You might think that's strange, because when I drink alcohol I get excited and happy and groovy, but you have to keep this in mind here. The way alcohol works is it inhibits part of your brain that does an inhibition. So you have part of your brains that says, don't say that the other people, keep your hands on, stop yelling, and alcohol basically inhibits the parts of the brain, making you more exuberant. Then over the course of this, in the course of drinking too much, it also inhibits other parts of the brain. So you could pass out and fall on the floor and at large enough doses die. So both curare and alcohol in different ways bring things down. Other drugs bring things up. So amphetamines, for instance, increase the amount of norepinephrine, which is another neurotransmitter that's responsible for genetic general arousal. That is how drugs like span or cocaine work. Other drugs like Prozac or L-Dopa influence neurotransmitters in ways that they increase, for instance, the supply of dopamine or serotonin, which can be relevant for uses like Parkinson's, which seems to be related to little dopamine, and depression, which is related to little serotonin. So these drugs work by influencing neurotransmitters, either by directly pumping in more neurotransmitters or increasing the supply in different ways, or stopping them from having effects by binding then offsetting them of different ways, but they work through their effects on neurotransmitters. So the more general idea is the way neurons lead to thinking is that they form clusters or networks. These clusters and networks are computational devices that do interesting things like recognize these faces, or waking up right, or understanding sentences, or doing math, or experiencing great sadness, or falling in love, and so on. We now know that that's possible because we create computing machines that work in certain ways. That if you wire up a computing machine in certain complicated ways, it can do mathematics, play chess, do phase simulation, and so on. So you may be interested in the project of computation neuroscience, which tries to ask the question, how are neurons wired up to do interesting things and use our own success at computation theory as a model. Then sometimes takes the inference the other way around, which is you can see how people do it, and they use the knowledge of how people do it to create computation system that can do it as well. So how is the brain wired up? Well, you might imagine that it's wired up like a portable computer, like a laptop, like the sort of computer you're looking at now. Into some regrets, it is, but there is a couple of reasons why it can be, and both of them have to do with how well the brain works. So first, the brain is highly resistant to damage. If you get a knife to the brain, if you get damage to the brain, it will typically shut down a whole system. The information and capacitors somehow distributes across neurons in such a way that makes them extremely resistant to damage. While in contact, somebody could open up the back of your laptop, put out a chip, and the whole thing is returned. The whole thing will stop working. But the brain is wired up in a certain way that makes it healthy resilient. The second thing is the brain is wired up in such a way that makes it work very fast. So computers can do millions of operations per second because they're purely electrical. But brain tissue is much slower and can spend the time to do many steps. So to put it in different ways, if your brain was wired up like a computer, it would be so slow as to be entirely unusable. It has to be wired up in a way that's more efficient, that allows for slowness of brain tissues and neurotransmitters, and can still compute things at a human level, which is often brilliantly fast. Because of this, there has been a huge interest in massivity parallel system and complicated neural networks, which are wired up as we believe the brain does, and as such, we're helping computers to do things based on our understanding of the brain that they could never do before. The The details of this is something we're going to talk about throughout the course. We're not actually going to end up explaining different capacities directly in terms of neurons because we can't, and because we want it to have higher level explanation. So when I talk about how people learn language or how do they recognize faces, we're not going to talk much about neurons or in particular, but we will talk about different brain areas and how they work. Then the assumption is, the bit is, that everything we talk about in more functional ways can ultimately reduce down to large networks of neural systems, and then its turn will ultimately reduce down to the specific behaviors of these specific neurons that we're looking at.